Our study dives into the intricate relationship between the blood-brain barrier and sleep regulation in Drosophila, particularly under the influence of a high-salt diet. The blood-brain barrier serves as a crucial gatekeeper regulating the exchange of substances between the bloodstream and the brain. Our investigation focuses on understanding how the blood-brain barrier may be implicated in the disruption of sleep patterns when subjected to a high-salt diet. Our research will be looking into how the permeability of the blood-brain barrier influences sleep patterns in Drosophila under the influence of a high-salt diet. We hypothesize that a high-salt diet affects permeability of the blood-brain barrier, which will decrease sleep, where minocycline will maintain the blood-brain barrier's permeability. To test this hypothesis, 1 to 3-day-old Drosophila were transferred into different food groups, including high-salt diet only, high-salt diet and high minocycline, normal food, and high minocycline only. Drosophila were then tracked under an ethoscope for a 24-hour period.
Our pilot data indicates that a high-salt diet increased mortality in Drosophila, which was mitigated by minocycline treatments. This diet seemed to reduce sleep, a trend prevented by high concentrations of minocycline. Although the high mortality rate complicates direct hypothesis testing, these findings highlight the complex relationship between the diet, the blood-brain barrier, and sleep regulation, emphasizing the need for further research. Additional replicates are required to strengthen our results and investigate the causes of increased mortality under a high-salt diet.
One limitation of our study is the need for additional replicates. We also need to explore potential reasons for a greater mortality rate in flies exposed to a high-salt diet. Future research endeavors should delve deeper into the underlying mechanisms through which disruptions in blood-brain barrier permeability influences sleep regulation.